Thanks very much for the invitation to talk to you a little bit about our work. So as you said, I'm Molly. I'm a postdoctoral researcher at Barts Cancer Institute, and that's part of the City of London Cancer Research UK Centre. And I'm part of Professor Sarah McClellan's laboratory, and we're predominantly interested in understanding chromosomal instability across many cancer types, and we're a fundamental research biology lab. And so, as I was saying actually to Tony over lunch, that we're quite patient removed. So actually hearing some of your stories today and meeting you and chatting with a few of you, it's actually quite a, it's really important to me, and it's made me feel passionate again about, why we do what we're doing. So thank you very much. And yes, so what I'm going to talk to you about today is a research project that we're very grateful was funded by GIST Cancer UK and Sarcoma UK, and I'll read it out to you again. So this is about chromosomal instability in genomic evolution of GIST and its implications on TKI therapies, which is quite the title, as Nick's suggested. So I'll hopefully try to explain some of that to you today as best as I can, but if anything's confusing, please raise your hand and ask questions. Okay, so I'm going to start with just the basics of the genome. So this is, there are trillions of cells in your body, and this could be normal cells, or this is maybe your tumour cells as well, and each of these cells, or most of these cells, have got a nucleus. And the nucleus is what contains all of the instructions for, your growth, your well-being of your body. And so these this nucleus contains the chromosomes, and these are structures that you can see a picture of here on the right. And these are contained in this nucleus, and each of these chromosomes is made up of this intertwined DNA, which is your genetic code, which is all the instructions for your cells to do everything that they need to do. So we're interested in the chromosomes, and this is actually a representation of your chromosomes and your genome. So if you do or don't know, there are 46 chromosomes in every single cell of your body, and these are split up into 23 different chromosomes, from 1 to 22, as well as the sex chromosome X, and these are 2 copies of each of these chromosomes in your body. And so we're really interested in when these chromosomes they go wrong, or more that they change in their number. So, for example, you may have heard of the disorder Down syndrome, which is actually caused by this increase in copy number of this chromosome 21, which in embryogenesis, this causes quite a lot of, as you can imagine, a lot of problems and a lot of defects into the individual. And so now I'm going to show you the karyotype, or the chromosomes of cancer. This isn't a GIST cell, this is just any cancer cell at the moment. And you can see that here. So if we start to look at each of the individual chromosomes, 1 to 22 and X, you can see that actually most of them are not present in the same copy number as they were originally here. So what we're really interested in understanding is how you go from having this normal karyotype, or normal number of chromosomes, to this karyotype here that you see, where the chromosomes are actually represented in a lot of different number. So this can be both gains of chromosomes as well as losses of chromosomes. And finally as well, chromosome instability isn't just the change in chromosome number, but also the structure of the chromosomes. So we can also see this represented in types of graphs a little bit like this. So this is a percentage copy number profile graph, where you have the chromosomes along the bottom from 1, 22 to X. And this is the percentage of GIST patients who have different copy number changes. So if you have the line going above the middle here, it means that it's commonly a gain of that chromosome region. And if you have it going below the line here, it's commonly a loss of that chromosomal region. So for example, around 45% of patients have a loss of this small portion of chromosome 1. And so to understand why this happens, we need to understand what's going on. So this is going to go back to maybe some biology you learned in school or college, or maybe you didn't learn it at all. And actually this might answer some of your questions about what mitosis is as well, that we had from earlier on in the meeting. So this is the life cycle of the cell, and it can be represented a little bit like this. It's broken down into different stages. So the cell usually begins in this first growth phase, where you have each of the chromosomes represented. It's doing its normal cellular function, and it's getting ready to double its genome, ready to make 2 new cells. So then we move into this next phase of the cell cycle, which is the DNA replication, where the DNA and the chromosomes are all doubled in number, so that they're represented. They've got double the amount of chromosomes, so that when you split the cell, you have enough of the genomic material for each of the new cells. Then we have this second growth phase, which is where the cell will have a pause. It will have a time to check the DNA, correct any errors that were made, as well as double all the other components of the cell ready to split into two. And that's when we move into the cell division. And this is actually what mitosis entails. So it's when the cell is dividing into two new cells. So in a normal cell, the chromosomes will align along the middle of the cell like this, and then they'll be dragged to either side of the cell. And then, I've put it nicely here, this is then split into two, just like this. Obviously, it doesn't happen like that actually in the process. There's no scissors involved. And then we split the cell into two, the outer membrane refuses, and we end up with these two new cells that have got each of the copies of the chromosomes. So how does this happen in cancer? And there's a bit of a spoiler here, which I didn't get rid of by accident. So we start in the same phase, in the first cell growth phase. We go into DNA replication. But DNA replication is different in cancer cells because of possibly UV exposure from the sun, or smoking, carcinogens, all these other things that happen in everyday life, and this causes DNA damage. So the chromosomes have become damaged, and it means that they're not replicated correctly and they have errors. So then we pass into the second growth phase, which is what I talked about earlier. The cell will stop, and it's when there's a period of time to correct the errors that happened during cell replication, DNA replication. And what would happen in a normal cell if there's errors or DNA damage is that they would stop there, the errors would be corrected, or the cell would die, because it's not a good cell to keep going forward and making new cells, so it will stop, it will die. Whereas with cancer cells, because we know that there are mutations that mean that they aren't checked or it's not corrected, they actually continue into this, into cell division or mitosis. So as before, the chromosomes align along the middle of the cell, and they go through the same division. However, a DNA damaged chromosome, such as this one stuck in the middle, isn't pulled apart to the end of the cell as the others are, and therefore the two cells that you get after cutting them apart, it means that one of the cells has got too few chromosomes and one of them has got too many chromosomes. And so therefore, we have two cells that are made at the end of this, and they have an unbalanced genome. They don't have all of the components they need to control the growth of the cell or promote growth of the cell. And we can actually see this happening in the lab. So this is a cancer cell actually going into that second growth phase, and we'll watch it a few times. I hope you can appreciate well, for me, this is quite a cool video to watch. But this is a cancer cell going into cell division. It's going to do it again. So the cells, the chromosomes become visible. They align along that middle plate of the cell, and then they divide to each end of the cell and create these two new cells. But hopefully you caught it in the middle of this division. There's a piece of the chromosome that doesn't actually travel to each end of the cell. And that means, as I said before, we've got two new cells that have been created, and they don't have the proper complement of the genome at the end. And so hopefully you can imagine that this is something that's a continual process. It's going on and on. Every organ of your body, there's millions of cell divisions happening all day, every day. And if you imagine that this is happening, for example, in the context of cancer, is that we end up having, this unbalanced genome, and it's a continual process. So actually, by the end of, if you have a whole population here, you'll end up having a load of cells that have all got a different number of chromosomes. It might be chromosomes that are represented in double the amount, or they're all just slightly different. And that means that actually, when we treat them with drug therapies like your TKIs, that they might actually respond quite differently to the treatments. So why do we care about chromosome instability? We analyse it in a number of different cancer types as well as GIST. And it means that the cancer cells have got a slightly different genome to normal cells. We know that from clinical studies that have shown that in patients who have got the incorrect genome or poor or different chromosome number, they have poorer outcomes. So you can see that represented on this graph on the right here. This shows the percentage of patients that have survived, and it's got the abnormal genome and the normal genome patients. represented in the two different lines. So if you look at the light blue line, you can see that actually by the time we get to 3,000 days, a lot less of the patients are kept surviving. They have a poorer outcome than those with a normal genome. And so what we also know is that chromosome instability means that each cancer cell as well has got a slightly different genome, which means they respond very differently to drug treatments, even within one tumour. So I probably don't have to introduce what GIST is to you, to this audience, but just in the context of our research, we know that there's been research that has shown that GIST tumours have some aneuploidy, or that cancer cells that have got too many or too few chromosomes, or that the pieces of the chromosomes are represented in different proportions. We know that treatment with TKI is actually very, very effective. And as I've heard a lot about today, is that a lot of patients respond well and they're on it for quite a long time, and it's very good. However, we also have heard that some patients don't respond, or the tumour comes back, and the cells that are now that have come back are now resistant to these TKI therapies. And so we know that chromosome instability in a broader research context is just one of the ways that a cell can become more adaptable, or more flexible to adapt to survive in different treatment circumstances. So what we want to know is if GIST tumours show signs of these chromosomally unstable phenotypes. And so this was a collaboration with a very talented clinician research scientist, Dr. Cesar Serrano, which you can see an image of here, and his PhD student, David, who are based at the VIHO Institute in Barcelona. And their research focus is completely on understanding GIST biology. And so our interest in chromosome instability and their background in GIST, we came together and tried to understand a little bit more what's going on. And so David, the PhD student, actually came to our lab to do some cell biology experiments. So in our labs, we use cell line models, which are cells that are derived from patient tumours that we grow in a dish like this. We do this because it's not as easy to get hold of patient material, but also it's very hard to do these cell biology experiments in patient material on patients. So we do it in cell line models here. And so to understand if there's chromosome instability, what we do is we look at cells going through that cell division that I explained to you earlier with a high resolution microscope. And so this is a cell going through cell division with the DNA or the chromosomes labelled in white at 100x zoom. So I think, like, if you imagine your iPhone goes to like 2x zoom, this is quite a lot of zoom. So we score how many cells each are going through this process and how many of them make an error. So on the right here, if we do this same experiment using normal cells, around 2% of the cells make these types of errors, and these are mostly corrected, or the cell will die, as I said before. But when we've done this in two GIST cell line models, we've noticed that in over 15% of the cases that we've scored, that they have these kind of cell division errors ongoing. So one of the other things that we said we wanted to understand is how this affects the genome evolution. And so evolution in this context might be a bit weird, because you expect evolution to be about, going from gorilla to man, but actually in cancer, evolution is quite a big concept, because you have this quick change in your genome over a short period of time. So we're really interested to see what actually impact this has on the genome. So we've just derived this kind of complicated experiment, which I'll try to explain to you. Because we know that the tumour at the beginning, if you remember that graph I showed you, graphic I showed you, where the chromosomes in each of the cells is all quite different. So we want to understand for each individual cell how they might behave differently and how their tumour evolves. So to do that, we take a single cell out of this mixed up population, because it's quite hard to, if you have like a mixed up population, to figure out what's actually changing, what the chromosomes are actually doing. So we take out one single cell, and if you can imagine that maybe this is a chromosomally stable cell or a normal cell, if we do this experiment, we grow them on their own, and the cell won't change, and you have this population at the end that are all quite similar to each other. But if we do this in a chromosomally unstable cell, what we're expecting to see is this kind of concept here. So we have this heterogeneous or mixed population at the beginning. We take one cell out, we allow it to go through cell division, and we end up, some of the cells create these errors. And then by the end of it, if we continue this process, the tumour population at the end is actually a big mixed up bag, and they all have different alterations. But because we've done it in this way, we know that the alterations that have happened at the population at the end, when we do our sequencing or our analysis, that it's something that's happened during that time in culture, and it's not some historic problem that has been in the cell for a long time. And so this is just some data that we've shown from some of the single cell sequencing that we've done at the end. And this is a similar graph that I showed you at the beginning, whereas that was percentage of patients that have got copy alterations. This is percentage of each single cell at the end population here that have got different chromosomal changes. And again, the chromosomes are at the bottom, and each peak at the top is chromosome gain, and each peak at the bottom is chromosome loss. And we were quite surprised that over just this short time that we've got all of these alterations in the genome already. So what were the actual main findings of our research so far? We have shown that chromosome instability does fuel tumour evolution in GIST, and we believe that it contributes to the aggressiveness of the GIST tumour. And we also believe that it enables the cells to become more adaptable and harder to treat. We also showed something that I haven't actually explained to you yet, that whole genome duplication is quite prevalent in GIST. And so this is a concept where the whole genome will be doubled because the cell has failed to go through that cell division, that mitosis. So actually it's got double the complement of chromosomes that it needs. So this means that when they actually lose one of the chromosomes, in comparison to a cell that's got just the normal complement, they have an extra backup copy. So they're able to tolerate more errors before they induce cell death. And so this is all great, it's all well to do, but what does that actually mean to do with the therapies that you have as patients? So we want to understand how chromosome instability is influenced by TKIs to help us understand why the cells become resistant and help us understand how we can treat those cells specifically when they come back. So to do this, we're going to do a similar cell experiment that I explained just before, where we take the single cell out of the population and we'll let one of the cells just go on their normal way and do their chromosomally unstable cell divisions. And we'll do this other treatment arm where we have the single cell isolated into TKIs, allow it to go through division, and sequence the population at the end, predominantly to understand what are the chromosome patterns that come out normally, as well as how do they differ from the ones that have been treated with TKIs, and do we actually pick out a pattern or a chromosomal complement that is different when it's a resistant cell, for example. So once we know the patterns, hopefully then we can understand more why they're resistant and be able to pick out in patient tumours what the resistant mechanisms are to help better treat the patient that's come back. Okay, and with that kind of complicated story, I hope I haven't lost you all, but if I have, I'm very sorry. I'd like to thank the people that are in my lab, particularly Sarah McClelland, who is obviously in charge of all this research, Isabel, who did some of the single cell work for us, and David, who came over from the WHO, and of course, the patients and families that contributed their cells to this work. Thank you to GIST Cancer UK, Sarcoma UK, and of course thank you all for listening. And if you are still with me and have any questions, then please shoot them off.